Hello, welcome to the second in the GeCIP and QMUL seminar series. We're really happy today to have Dr. Dennis Wang and Dr. Jamie Ellingford. Dennis Wang is talking first. He did his Masters and PhD in Cambridge and then a postdoc in Toronto. He then worked at AstraZeneca for a little while in oncology drug development. And he's now a lecturer at the University of Sheffield. And he belongs to the GeCIP domains education, renal cancer, neurology, and machine learning. Thank you, Dennis.
Thanks, Anna. Thank you for having me here. And so I'm at Sheffield as a lecturer and group leader in genomic medicine. So that means I give lectures on genomic medicine to current and future NHS staff, and I also do research mainly in the area of pharmacogenomics using machine learning methods. And I'm not actively involved in all of those GeCIP domains, mainly because I'm kind of applying machine learning methods across kind of disease domains. I have maybe I get invited to some GeCIP domains to scope out if there's any kind of methods that could be applied to a specific disease. Okay. But in this talk, I'll kind of give a little bit of kind of preliminary results that we generated from analyzing the whole genome data, not linking it to any specific clinical phenotypes, but just looking at kind of the genomic features across some of a small sample of the 100K data. Okay. So a little bit about where I'm working currently. So it's translational. I'm currently based at the Institute for Translational Neuroscience, and it's a translational institute.
Can I stop you briefly?
Okay, sorry. So where I'm based in Sheffield is encompassed by a biomedical research center funded by NIHR that basically translates research from the lab to the clinical setting and encompasses the hospital, wet labs, people doing kind of basic biology, and also a lot of people who are doing engineering and applying kind of biomedical engineering techniques to a lot of the patient samples that we have. Okay. So I'm part of a bioinformatics group in the Faculty of Medicine, and basically we're probably the only bioinformatics group in the whole medical school. So we get involved in a lot of kind of projects in different disease areas. We have some functionalities that's common to kind of like a core service, but we're also utilized in kind of providing bioinformatics on clinical samples, working with the Sheffield teaching hospitals, designing NGS experiments for sequencing that's done at the hospitals. And we also provide some training to the clinicians there, to some of the researchers there on how to analyze genomic data, and just some basic bioinformatics, training on some basic bioinformatics techniques. But most of us, I think, come from a purely more academic research background or translational research background, where a lot of the bioinformatics is hypotheses driven. So this is kind of an example of where we're starting from a kind of basic question around maybe a cellular mechanism, and we're designing a RNA sequencing experiment to understand a specific mechanism in a cell. And then we're building a bioinformatics pipeline, statistical models to interrogate the data, visualize the data, and test that hypothesis or provide some biological insight, right? And then we go back to kind of the bench scientist to validate some of the findings, right? But we found in some of the work we do with the Sheffield Teaching Hospital, The clinical kind of pathway for bioinformatics is a little bit different. It's not so much, it's a little bit more data-driven, not so much starting from specific hypotheses. There we're getting a sample, doing sequencing on it, and doing some kind of doing more discovery work and kind of parsing the data, identifying variants that may be pathogenic and clinically actionable, and helping clinical scientists produce reports that can feed back to the clinician and inform, say, treatment plans for the patient. Okay, so that's also a linear pathway as well. But we found the difference between those kind of pathways is that in a research setting, we actually try various experimental pipelines, right? So, for instance, if we're doing an NGX experiment for a pure academic study, we might try a set of different variant colors, produce different results, and look at the overlap and interpret the results. But I think in a clinical setting, it needs to be kind of more of a fixed pipeline, and we need to kind of develop best practices so that the results that we provide are consistent over multiple patients. Right? So that's kind of a slight difference in how we conduct bioinformatics as a service in the research versus the clinic. So that involves us to do more documentation, ensure more provenance in the data, and do more quality control. And this is what we're trying to learn from the 100,000 Genome Project, is kind of go through some of this kind of clinical best practices for bioinformatics. Okay. And the example I'll give next is how a case where we're trying to do some quality control on some of the human genome sequence data. So first, my research group began working on cancer and looking at kind of the mutational spectrum across cancer. In particular, we're looking at the mutation burden across cancer. Right? So this is an example of a review paper, where they show kind of the rate of mutations across cancers, and it's quite variable. And you can also build whole genome signatures from the data and relate it to particular phenotypes, right? So that got us interested in looking at a specific cohort. So we collaborated with Princess Margaret Hospital in Toronto, where they were specifically interested in non-small cell lung cancer cases. And there, we looked at essentially the mutation burden across patients. So on the left here is an example of around 36 patients ordered by the number of point mutations and copy number aberrations. Okay, and then we can relate it to a particular phenotype. On the x-axis is disease-free survival years, and we generally see kind of particular pattern, right? And then we will fit a statistical model or machine learning model to that data to select features that may be more associated with the phenotype, like survival, right? And the overall aim of that is to stratify patients based on those genomic features. And hopefully the different groups of patients will correlate to kind of different patient outcomes. Okay. So we did that. Our model kind of detected a set of 865 genes that were able to kind of stratify patients based on the mutation burden. So the mutation burden here is called NAG. It's just number of altered genes. So we're counting copy number and point mutations here. And it basically divided into three groups of high NAG, low NAG, and a group where there's high number of altered genes and immunogenic mutations. Okay, And then, so we found that the group with immunogenic mutations and high mutation burden seems to be, seems to live longer for early stage non-small cell lung cancer. Okay? Whereas patients with a low number of mutation, low number of altered genes have a poor survival. Okay? And the rationale for this, we think, is that the high number of mutations may be generating more immunogenic peptides that may activate the immune system to attack the tumor. And then we can see from the RNA-seq data that the group with the high number of alterations have higher expression of cytotoxic T cell markers. Right? So this is just an example where we kind of used the whole genome data to identify features related to a phenotype, right, and developed a kind of prognostic biomarker, right, that may be predictive of survival. Okay? But when we analyze, how stable is this prognostic biomarker, we found that it really depends on how we actually count the variants, right? So we determined that the set of genes had variants that may be prognostic, but we have to ask the question, what variants, are those variants real, and what was the metrics we used to identify the variants? So say if we were really to kind of bring this prognostic gene panel to a clinic, and someone else does sequencing on a different cohort of patients, will they use the same metrics to identify the variants? And obviously, one of the metrics is the allele fraction of the variant, and there's also kind of other metrics that you can use, like the quality score, the mapping score, and kind of properties of the variant, whether it's somatic, protein coding, non-synonymous, and so forth. But in this case, we focused more on the allele fraction and analyzing to look at basically to look at what allele fraction is sufficient for us to identify that as a variant that may be prognostic. Okay. But we know from other studies from ICGC that the variant calling can be there could be low concordance between the variants that are called. Right. So this is just a table showing where ICGC, I think they sent neuroblastoma samples to different labs, and then so the same sample, same data, and then they asked different labs to call the variants, and this kind of the level of concordance across variants, right? And there could be low concordance between mutation calls where there's low allele fractions, right? But you could have higher concordance if you obviously combine the variant calls. But generally, there was kind of no best practice as what limit for allele fraction to use or what variant calls to combine. Mm-hmm. So we decided to, instead of kind of looking at the algorithms and looking at how they interpreted how they called the variants, we decided to look at the allele fractions more closely, and we want to answer the question: what is the minimum allele fraction for a real variant? First, in the germline, because I think in the somatic case, it's a little bit more tricky because there could be subclonal mutations, right? So more basic question, we decided to focus just on the germline. What is the minimum allele fraction? And our hypothesis was that it depends on the genomic loci. So we don't think there's a kind of a global limit, and that some loci may have consistently low allele frequency across individuals, across different diseases as well. Okay. So this is a kind of signal to noise problem. And traditionally, you look at this problem sample by sample, right? So you check, what is the allele frequency in regions where you don't expect variance, where you do expect variance. So you look at the allele fractions on a sample-to-sample case. But in this case, with the 100K data, and also in a lot of kind of clinical samples, you don't want to look at each sample individually. So we try to see if we can assess this problem from a population, from a group of patient samples. Okay. So basically, without de-anonymizing without identifying each sample, can we assess kind of the allele fractions? So this is just an example whereby there's 4 patients, and what we do is instead of looking at kind of the allele fraction in each patient at a particular locus, we look at the fraction across those 4 patients, right? So for the example of a particular locus where there could be the A allele called, right? The allele fraction across the 4 patients would be 37%, right? But this 37% could be a composition of homozygous and heterozygous mutations, or cases of no mutations, no variant alleles, right? But if you have a composition of both homozygous and heterozygous variants, you would get a specific allele fraction with a high standard deviation. Okay? But if it was a consistently low allele fraction, like the example of a C, right? You would get a low allele fraction, but also a low standard deviation. So this is kind of the case we're trying to pick up, is that we're trying to identify, without looking at each individual patient, just from the summary statistic, we're trying to assess, which variant alleles are consistently called at a low allele fraction. Okay, similar to the C case. Okay. Obviously, there are cases where there's sequencing errors, like the case of T, right? Where there'll be you'll always get kind of a low allele fraction, but it will kind of fall below a certain threshold. It will have a low standard deviation, but that's usually kind of sequencing error. So we're actually looking at kind of called variants that are still at a low allele frequency, but kind of consistently across patients. Okay, that's kind of, okay. So we did this first on 150 patient samples from a collaborator in San Francisco in Personalis, and we observed that there is, if we plot the allele fraction and the standard deviation across patients, we observed that there is a group of variants with low allele fraction and low standard deviation. And this is in comparison to an expected model which we generated based on mixtures of individuals with like homozygous and heterozygous mutations. Okay, and basically this group of allele variants with low allele fraction and low standard deviation, we termed it as kind of suspect loci because they kind of deviate from the expected model. Okay, and then we validated this on the 100K data. So this is just a plot on chromosome 21. We're still working on exporting the data results from the other chromosomes, but it's taking a little long. But we use basically patients from the neurology samples, and we also kind of identified called variants that were consistently low allele fraction and low standard deviation. Okay. And these suspect loci seem to be at a high proportion in regions that are GC rich or repeat rich. Okay, and as expected, they're at a low proportion in the NIST high confidence regions. Okay, in both the Personalis cohort of 150 patients and a small group of patients from the neurology domain in the 100K data. And then we also looked into the reference material, so the genome in the bottle, and found that those suspect loci were also at low allele fraction in the genome in the bottle. So it's kind of independent validation on kind of a high quality genome where these suspect loci are actually indeed at low allele frequency. We don't know why, if that is due to some kind of sequencing artifact, or it might be something to do with the structure of the genome. But this is still an open question for us. So the relevance of this is that we also looked at some gene panels, and we also found different proportions of suspect loci in the gene panels. There were differences between exons and introns, which is quite interesting. We're looking into that. And then there were also differences between cancer gene panels and other neurological panels of neurological disorders. So we found that there were high proportion of these suspect loci in the cancer gene panels compared to this intellectual disability panel that we found in panel app, consisting of about 2,000 genes. And because in 100K data we used mostly neurology domain samples, and most of the patients had like kind of intellectual disabilities. Right? But this is something to, I guess, be aware of. I think the possible explanation for a higher proportion of suspect loci in the cancer panels is that maybe in the cancer panels they were determined as kind of cancer-associated genes because people found low allele frequency, mutations of low allele frequency in those genes, and they might have interpreted them as like subclonal mutations. But I think, our results show that they could also be kind of maybe sequencing artifacts, but we don't know, right? So there's a higher chance of, confusing subclonal mutations with kind of sequencing artifacts, maybe in the cancer genes. Okay, so that's just a kind of preliminary analysis that we're taking forward. We're going to scale it up to more patients in the 100K data, looking not only at the neurology domain, but some other patients in the other domains. Okay, and we're going to also try to see if what's the reason behind these suspect loci, whether there are issues with actually the sequencing protocols, the sample preps that may be causing the suspect loci. So the other work we're doing is actually more kind of disease related. We're leaning on this kind of landscaping paper for the bladder cancer, where we're actually probably going to use some of the panels and panel app to kind of validate the different subtypes of lung cancer in bladder cancer that have been found in TCGA. But we're going to especially focus more on structural variations and non-coding SNVs that take advantage of the whole genome data. So far, what we found is that there still requires more QC to be done on the structural variants that have been called by Genomics England. And we're still trying to figure out a process whereby we can validate the SNVs, so whether we can actually recall some of the samples and do some functional studies on the SNVs. And also, we found that the clinical phenotypes in the bladder cancer cases, the clinical data in the cancer bladder cases is quite sparse. And it's not that the data isn't in there, but I think a lot of the annotation, a lot of the information is still encoded in the free text fields. And we're recruiting clinicians to help us kind of do further annotation on some of the clinical variables and kind of, and we'll also need some more follow-up on the patients to get kind of better survival data once we find frequent, recurrent genomic alterations that we want to correlate. Okay. So finally I'd like to thank some of my collaborators in Toronto, in Personalis, and the team in Sheffield. Okay, and another plug I want to make is that we formed this Northern Bioinformatics user group to kind of discuss kind of genomic analysis issues with the 100K data and also other data. Jamie gave a talk there at the first meeting in Sheffield, and the next meeting is at Newcastle in January. So there's no strict boundary for what's Northern, so anyone can really attend if you get up there to Newcastle. Okay. So I mean, this kind of more venue that we can talk about more some of the technical issues, best practices, for variant calling genomic analysis and so forth. Okay, thank you.
Thank you very much. Have we got any questions? Yeah.
Sorry, I might have missed it because I was late to your talk. Did you see any difference in the mapping scores of your low allelic fractions? Did you see any difference in the mapping scores of your low allelic fractions versus norms?
Yeah, we didn't see, there's a slight difference, but we didn't see really a significant difference there. Because, I mean, we were taking variants that have been called already, so there was kind of some quality control on the mapping scores. But we do want to see, yeah, with just without calling variants, just from like kind of the pile up of the reads, whether there might be some difference. I think, I mean, there obviously is because there is obviously some difference because we do see more suspect loci in these GC-rich regions and regions that are difficult to map, right?
I'm just wondering, did you see any differences between build 37 and 38 in terms of the suspect loci? Did you do that comparison?
Yeah, we initially took both 38 and 37, but it turned out we can't really look at the two. So actually the analysis showed was just on the 37, and also the analysis on the 150 healthy cases was on 37. But yeah, I mean, that is something we could do. Yeah, once, I guess, because I think when we looked at it, not so many cases were realigned to 38 yet.
Can I ask a question? Maybe it's a stupid question, but are you only using blood as your tissue, somatic, germline tissue? Could these be somatic mutations in blood cells? Have you compared to saliva, for example, to see if they're still there?
No, we haven't looked at the samples. Yeah, I think the 150 cases are mostly in blood, but yeah, I mean that is the difference between samples is definitely something we want to compare, right? Whether there is a relationship with the quantity of the DNA and so forth. But yeah, both cases were both from blood samples.
So when we were looking at CMVs in samples extracted from saliva versus blood, there was a big difference in some common mutations between them. So.
Yeah. Well, I mean, in this analysis with the suspect loci, we didn't look at CMVs. It was just purely on the point mutations. Yeah, definitely.
Can you use databases such as Nomad, because they'd give alternative value frequencies for in a really big data set, and that may help kind of understand some of the sequencing technologies. If you tried looking at these variants in there to see whether they also have low frequencies of these variants as you see them in these samples.
Yeah, we haven't looked at Nomad extensively. We did look at. One thing we did look at is comparison of kind of common and rare variants from Nomad, and it's kind of too early to say. We did see some differences, but I think we need further validation. But we are looking into that. Yeah.
Yeah, it's a useful point about the finding it in blood versus saliva. Because these variants that are enriched in GeCIP, clonal hematopoiesis, in marrow, they'll concentrate in cancer genes because for the very reason they cause a growth advantage in the marrow. Yeah, they go up in frequency with age, and they're commoner in people who get cancer. Yeah, for whatever reason, either it causes it or it's due to the same things that have exposed the patient to get cancer. So there's a lot of contamination in Nomad of alleles that are just too common to be pathogenic, but that's the very reason they are. So when you look at the normal distribution of the calls in a normal population, you get a lovely curve, but the ones subject to GeCIP are just a straight line across. So you can actually tell them apart. And, credit to Myriad, I've actually been through their database to sort out the ones which are the ones which probably are chips versus the Gaussian distributed ones that come down at a good score. So that might account for some of them, and of course because you've been looking in cancer patients, that's where you'd expect to find them. Yeah, just a thought.
Okay, well, let's thank Dennis again.
Okay.
Thank you very much.
Thank you.
So now I'm going to introduce Jamie Ellingford. Jamie has been based at the Manchester Centre for Genomic Medicine since 2012 and is currently a research fellow funded by the Health Education England Genomics Education Programme. He did his BSc in biology at the University of Sheffield before tackling an MRes in translational medicine and a PhD in genomics at the University of Manchester. His PhD focused on informatics strategies to analyse and interpret big genomic data sets in the context of rare disease, which is good for us, with a major emphasis on rare conditions impacting eyesight. His PhD led to publications in The Lancet and leading clinical genetics journals. And Jamie's big questions are evolutionary genomics, applied omics in healthcare, and his current work is combining these interests to develop and apply bioinformatics and wet lab approaches to identify pathogenic variants in the non-coding genome.
Thank you very much.
Thank you.
Is that okay? Sound, yeah? Perfect. Thank you all for coming along to see this. And there's lots of other exciting things to do in London at this time of night on a Thursday. So I'm based at the Manchester Centre for Genomic Medicine, which is one of the national genomic medicine centres and has recently become a genomics laboratory hub. So we're one of the centres in the UK that gets the first eyes on sort of gel data going through the genome medicine centres. I always rush this at the end, so I wanted to say thank you first to a lot of the guys that have helped with this sort of analysis, in particular Graham and Bill and Ray and Hugh in Manchester, and the guys at Genomics England that have dealt with my barrage of emails over the past year, in particular Freya and Damien and Chris. And Gavin, who sat in the audience here as well, for the ongoing work that we've got going with some guys down here at UCL. So what I'm going to talk about is sort of our experience in Manchester of dealing with diagnostic gene panels, some of the lessons that we've learned from dealing with these big cohorts of individuals for rare disease, and how we've applied these things that we've learned to the gel data. In particular, how we've looked at a type of cohort-wide analysis for the pilot data that's come through gel, give you some examples of this analysis and how we've identified disease-causing non-coding variants, and in the process talk about some of the bioinformatic and wet lab strategies that have been required for us to develop and apply to achieve this. So it's worth re-emphasising, I'm sure all of you already know this, but the genome is a large and complex molecule. It's comprised of 3.5 billion nucleotides, most of which we don't really know what it does, the 98% which is non-coding or repetitive. There's big international consortiums that are trying to work out, well, what's the function of these non-coding regions, including ONCODE or GTEx. But actually, the 100,000 Genomes Project offers a unique opportunity to understand how these regions are disturbed, can disrupt gene expression and regulation, and can cause disease. So one of the key principles of that is that splicing. We have a pre-messenger RNA molecule, which is turned into messenger RNA. And that process requires excising the introns from the pre-messenger RNA. And there's a load of different motifs within a gene that are required for that process to occur properly. So we understand a lot about how sort of these donor and acceptor sites, these dinucleotide regions that are immediately proximal to exons, how they can be disrupted and cause disease. We're starting to understand a bit more about how things like the polypyrimidine tract and branch points, how they could be disrupted and cause changes in gene splicing. And there's also the possibility to activate cryptic new splice donor or splice acceptor sites. And so I'll tell you a bit about some of these that we're starting to find, and also how we're starting to understand how the genomic context nearby to an exon can disrupt gene expression. So our diagnostic panels, at Manchester there's lots of different panels that are offered. This one's my favourite, which is for inherited retinal disease. So this is a set of disorders that is caused by mutations in genes which function in the back of the eye, in the light-sensitive film of the retina. Manchester for years has offered a 105 gene panel service which looks for coding plus or minus 5 bases mutations within genes known as the cause of these diseases. And they do quite well. We find a diagnosis for 50% 2 years ago, probably more like 60 or 70% today for people who are referred for this particular test. There's lots of different genes which are mutated. So of the 105 genes, over half of them were found to have a disease-causing mutation. And right at the end, I hope to show you why it's really important that for each patient that's referred, we can define exactly which gene is mutated. And there's all sorts of different types of mutations that we see, in particular missense mutations and nonsense mutations. Now, of note, there's a couple of things which are missing from there. So we look at splice region variants. These are 5 bases into the introns. And some known, well-characterised intronic variants. But these variants aren't surveyed systematically, and I'll show you why in a couple of slides. But 2 of the really interesting trends that we're able to see when we do a cohort-wide analysis of this data is that we have, firstly, an enrichment of carriers who are unsolved, who don't have a genetic diagnosis, compared to those who are solved. So carriers are individuals who have a single mutation, a heterozygous mutation, in a gene known as a recessive cause of disease, where both copies have to be mutated for the disease to present. The second really interesting trend is when we plot along the horizontal axes here the years that genes were first identified as a cause of disease, we see with the red line that we're increasingly identifying new disease genes, and these are becoming part of the service. But the blue line shows this asymptotic trend whereby the genes that we're discovering today are accounting for less and less diagnoses in the population than the ones we discovered 10 years ago, which sort of makes sense. But the clear learning message that I took from that was, well, if we can look at the genes that we already know about more comprehensively, and the 100,000 Genomes Project offers an opportunity to do that, then we might have better, more increased yields in diagnostic services. So why is the genome better? Well, I'm sure lots of you already know this, but these are the coverage profiles of sequencing reads that we generate for a gene panel versus whole genome sequencing. So the gene panel focused on the coding exons, about 50 bases either side, a genome, the whole genomic context. And that allows us to detect things like copy number variants better, but it also allows this opportunity to look at intronic variants in a comprehensive way. Finally, it seems like a long introduction, but how are we doing in GEL? So as part of the pilot analysis, there's been almost 5,000 individuals who've been recruited from over 2,000 different families. And the first tier analysis that all of these patients go through is to look at a set of genes selected through panel app and look at the coding and the nearby intronic regions of those genes for disease-causing variants. And for the answers that have gone back so far, there's a diagnosis for about 20%, which is great. There's a diagnosis for these people that they might not have got through any other means. But still, there's 80% of these people with apparently rare disease that haven't got a genetic diagnosis that could have one. So that's what my research focuses on. How can we find answers for these guys that haven't got a diagnosis through this first tier process? Of course, the numbers of diagnoses differs between different disorders. So we took a very specific hypothesis into the data available in the research embassy, and it's that we knew that these unsolved carriers would exist, so people with single heterozygous variants that were known to be pathogenic in recessive genes, genes where disease only presents when both copies are mutated. And we tried to assess, well, can we identify a rare non-coding variant that impacts that same gene that's in trans on the other allele in that particular patient? And there's a couple of streams to doing this. So the first is that we can identify any potentially pathogenic mutation as this first single hit. That's quite an ad hoc strategy and actually requires on either a clinician coming to us and saying, well, we know about this patient, this gene is highly likely to cause this disease, can you find the second hit? And if we apply this sort of strategy for missense variants, for synonymous variants across a cohort, it's actually highly susceptible to false positives. But we've got access to this tremendous data resource with thousands of individuals without a diagnosis. So we thought, well, we could do a bit better than that, and we can home in on a particular class of variants. So these are loss of function variants, so nonsense, frame shifts, and we can automate the analysis to pick out individuals who have heterozygous loss of function mutations in genes that are known as the cause of their disease and when they don't have a diagnosis. So when we apply this across the whole pilot cohort, we identify at least 80 individuals that fit this profile. They have a heterozygous loss of function mutation, and they've been unsolved through this first tier analysis. So then we ask the question, well, are there any single nucleotide variants within the genomic regions of these genes? Once they've been identified, we compare it to population databases. So firstly to GNOMAD, to understand whether there's rare variants, and to eliminate variants where there's homozygous instances present, and then within our own cohort to eliminate false positives to the GEL pilot. And when we do this for these 80 individuals, we see that over half of them have a rare or novel non-coding single nucleotide variant within the same gene. We can then start to ask lots of more interesting questions. Well, what's the phase of these variants? Are they in trans or are they in cis? What do computational tools say about this non-coding variant? Is it likely to affect splicing or expression? Does it intersect with regions that have been characterized by ONCODE or GTEx or the Washington Epigenome Project to be functionally relevant for the expression of this gene? And by integrating these resources and these tools into the research environment, we can start to build up a bigger picture and provide some evidence to the pathogenicity of these non-coding variants. So I promised you a couple of examples. This is one of the first. So cystic fibrosis, we've known the genetic basis for that since the 1990s, early 1990s. The individual who I'm going to talk about now, she received a diagnosis of cystic fibrosis in the early 1990s and was identified to have the most common mutation in the CFTR gene in 1994. It was in a heterozygous state. She was a carrier of a cystic fibrosis mutation. It was only through the Genomics England programme that we were able to identify a deeply intronic variant in CFTR, which was predicted to introduce this new cryptic splice acceptor site. So that wasn't enough. We wanted to actually prove that. So we looked at lymphoblast cell lines and looked at the RNA that which had been expressed from the control versus the patient who was carrying this mutation. And we see that the patient, it was expressed in 2 distinct copies of CFTR, one which was slightly larger than the other. And when we cut these out and sequence them, we see that this larger band contains a cryptic exon, which had been introduced as a result of this new acceptor site. So we recently made these findings available through bioRxiv, and if you go to my Twitter page, it's the pinned tweet on there, and there's a link straight to the paper. In a second similar example, this was an individual, again with a respiratory disorder called primary ciliary dyskinesia. She had a heterozygous mutation in a gene known as recessive cause of this condition. This was a nonsense mutation. We looked on the other allele. We found a deeply intronic variant in the same gene, which is having a similar effect. You were seeing 2 distinct profiles being expressed for DNA H11 and the inclusion of part of the intron in the mature messenger RNA transcript. So that's great. We can identify these things and we can prove that they're disrupting the way that the gene is being expressed, providing we can access the RNA. Now, we wanted to see, well, how many genes could we actually do that for? And so we took all the genes in panel up and looked at their expression profiles within the GTEx dataset. And what we see is, well, not a lot of what is in panel app is expressed in blood. And even if it is, it might not be the right isoform of that gene. So less than 25% has a relative expression value of which we think is probably sufficient to be able to survey these sorts of things. So if we want to assess this thing across the whole cohort, then we need to develop in vitro RNA assays. And so with a group in Manchester, in particular with Ray O'Keefe's lab, we started to develop that and apply that to findings that we're getting back from Genomics, from the 100,000 Genomes Project. So this is a mini gene assay. And what we do is we develop a splicing vector. The splicing vector has control exons in it, which we know will always be expressed. And then in between these exons, we insert a fragment of interest. And that fragment can be probably up to about 10 kb. Here I'm just showing you a single exon which is being introduced with an intronic variant near to it. We can then transfect these into a cell of choice. We usually use hexels, and then assess the RNA which is being expressed at the end. And you can see all sorts of different things being expressed. So here we see a wild type allele, exon being skipped, and an intron being included. Okay, well, why does that matter? So PDE6B, this is a gene which isn't expressed in blood, only expressed in the back of the eye. And this is an individual who's been referred with retinitis pigmentosa. I'm quite nervous seeing all the ophthalmologists in the room over there, but if you look at the back of the eye, and you can see there, this pigment, these bone spicule pigments shouldn't be there, and it's a sign of a disorder called retinitis pigmentosa. And hopefully the ophthalmologist will agree with me that that looks like a fairly mild form of RP, which could be in keeping with mutations in PDE6B, a recessive cause of retinitis pigmentosa. When we look on the other allele, we find not a deeply intronic mutation, but an intronic mutation nonetheless. There's a couple of things that we can tell from the computational simulations. First of all, we don't seem to affect the canonical splice site. There doesn't seem to be a huge difference in the weight that these scores give to the fact that a canonical splice site exists there. And this site, whilst strong in a wild-type allele and diminished there, probably isn't having an effect in a wild-type anyway. So whilst there's not a huge amount of evidence here that this could be disrupting splicing, but we still wanted to assess it given the phenotype-genotype correlation. So we did one of these mini-gene assays, and we inserted 3 exons with the variant here closest by to the middle exon. And we saw a very different RNA expression profile between the wild-type vector and a vector with this mutant in it. We saw 2 bands, one which was slightly smaller and one which was slightly bigger than it was expected of the wild-type. And again, when we cut these out and sequence them, we see that the wild-type aligns perfectly here with the PDE6B. You can see the blue transcripts here. Aligns perfectly with the exons in PDE6B. Whereas the variant in red has all sorts of things going on. So you get exon skipping, you get a bit of intron inclusion. So we're fairly confident this mutation is disrupting the normal expression of PDE6B and is acting as a loss of function allele and potentially as a cause of disease in this patient. So I want to take a step back, and I said at the beginning that hopefully I'll convince you as to why we need to know what gene is mutated in people with these inherited retinal diseases. So this was a young lad who was first seen by a paediatric ophthalmologist when he was about 2 or 3 months of age. He was shown to have roving eye movements. Electrophysiology at the back of his eye showed there was no response to light at all. So he was diagnosed with an infantile onset retinal dystrophy. The 105 gene panel diagnostic test concluded that they couldn't find a genetic cause for his condition. We then performed whole exome sequencing a year later and identified a homozygous mutation in a gene called IQCB1. So this is a gene which is involved in the assembly and the function of primary cilia. So primary cilia connect the back of the photoreceptor, sort of the outer segment of a photoreceptor with the inner segment, pivotal for its function, but also has functions elsewhere in the body, so in particular in kidney cells, and involved in the sort of the transport of molecules across membranes. So mutations in IQCB1 can be associated and are often associated with renal failure. So this young lad was referred straight to a paediatrician and a kidney specialist, and they started asking the right questions, and it was shown that his kidneys were going off fairly quickly. At this point, he was about 12, 13 years of age, and all these findings were consistent with nephronophthisis and a diagnosis of something called senior-Loken syndrome, of which infantile onset retinal dystrophy is the first presenting sign. So he was put straight onto hemodialysis. He received a successful kidney transplant, and importantly, that kidney transplant was before acute presentation. So end-stage renal failure, which can be going to hospital in the back of an ambulance. So in this case, an early diagnosis was really, really important. And for people who were coming in with these sorts of symptoms, identifying the gene, which is causal, is really important. So when we saw a similar case, which has been recruited as part of GEL, we were really interested in knowing whether there was potentially a syndromic cause of this disease and whether there needed to be lifelong management of this patient. So this is a similar individual. He was the only affected individual in his family, and again, he showed subnormal responses to light at a very, very early age. Through the 105 gene panel, he didn't receive a genetic diagnosis, but we identified that he was a carrier of a potentially pathogenic mutation in a gene called GeCIP2D. So GeCIP2D is associated with these infantile onset retinal dystrophies, but importantly doesn't have a syndromic effect. Earlier this year, through the first tier analysis of the 100,000 genomes data, he also didn't receive a genetic diagnosis, but we were able to look at, so the first thing we wanted to do was absolutely confirm that this was a pathogenic mutation. We developed one of these mini gene assays, this time with just 2 exons being inserted, and again showed that the mutant had a very different expression profile to the wild type. We looked a bit further and identified a really interesting variant which is upstream of GeCIP2D in an open region of chromatin. So we know that DNA has to wind tightly around histones to become part of every nucleus. And in order for genes to be switched on, that winding has to uncoil a bit for transcription factors and other machinery to actually access the DNA. So we can characterize these regions through sort of surveying for open regions of chromatin. This was the nearest to the start site of GeCIP2D. We hypothesize that this is a regulatory region. It's rare within the general population. It's present in multiple families who have phenotypic presentations which are stereotypical of GeCIP2D, and we're currently undergoing other in vitro assays to actually prove that this is a pathogenic variant. Well, how could it be pathogenic? Recently, we've shown that mutations like this are associated with hypermethylation, over-methylation of sites just upstream of genes, which can cause that allele to be switched off. So we proved this recently in a paper in the American Journal of Human Genetics, that when a mutation in the 5' UTR, potentially in a regulatory site upstream of BRCA1, that this was associated with switching off of that allele. And we showed that by showing that there was a heterozygous variant in DNA, and that allele that this was present on wasn't present in the complementary DNA derived from the messenger RNA. So in conclusion, we've started to show that we can identify these non-coding pathogenic variants through applying this carrier analysis framework. And the types of candidates that we're picking out include deeply intronic variants as well as regulatory region variants. This is applicable to both monogenic and heterogeneous rare disorders. Hopefully I've given you an overview of some of the bioinformatics strategies that we've developed to apply to this data and the requirement that we need to actually assess the effects of these things. That being RNA isn't going to be accessible for all the genes that we're interested in, and so we need to develop these in vitro functional assays as sort of a routine service within genome medicine centres, I think. So with that, thank you very much for all your attention, and again, thanks for everyone who's contributed to this project. Happy to take any questions.
Thank you very much, Jamie. And I have to say that hearing both of these talks today is really inspiring, because we're in the GeCIP office running the day-to-day sort of tasks of making sure people can do research, and you do sort of worry that things are being left behind, and just seeing, you're working on non-coding regions, and you're working on these strange variants, and it's really encouraging and I think very exciting, and also both of you doing things that will create a resource for other people. So thank you. Questions? Tim.
So you're spotting variants within genes in the non-coding, and how many cases do you see one and actually you decide it's not going to cause a splicing? I mean, how good's your bioinformatics in terms of filtering out false positives?
So we can start to put numbers on that. So from the 80 that we started with, they're predicted to be loss of function, and there's all sorts of levels of information that you can add to that to get to a truer truth, if you like. So are we using the right transcript, for example?
Yeah, that's another thing. Yeah.
And so we can reduce those numbers that way. If we've got trio information, can we do desegregation? Can we just pinpoint the ones that are in trans? If we've got computational predictions, I hope I don't offend anyone by saying this, but what we've got at the moment isn't up to standard. We can start to use things like Human Splicing Finder and MaxENT, but they're okay, and we can get a feel for things, but they're never going to be sort of you'll go to. I can certainly tell you how many things that we've taken to the lab that we're fairly confident in and haven't found an answer, and that's probably about 20%.
Right.
But even then, I don't believe so. With Gavin, we've got a really interesting one, which is a deeply intronic variant in a transcription factor key core motif. We've done these splicing assays, and actually we probably didn't expect to find anything different from a wild type, and we didn't. And that's probably because we're not doing the right assay. We need something which assesses in the right cell type whether that transcription factor can still bind to that region or not. So even for the ones that were wrong, I'm not convinced that we're wrong. But we've only taken about 15 to 20 to the lab.
But of course, the problem for confirmation is, of course, you can do it outside by mutating something and seeing if you can generate an effect. But you really need the RNA, and of course, the RNA isn't always accessible. But I smell a prediction competition coming on where, of course, we can possibly get at that RNA and do some blind tests with people trying to work out making predictions, and then we can assess them afterwards.
Mm-hmm.
Nice. That was a great talk. I thought you made a really good case for mini-genes. Do you think there are any other ways of getting at different kinds of transcripts, like stem cells or organoids or anything like that, that might have promise in this area?
Potentially. So the two that I showed you, which we'd sort of marked as the easy ones, so one was from blood. DNAH11 was straight from whole blood. The CFTR one we did from lymphoblast cell lines. There's other things that are accessible, like hair follicles, things like that. You could potentially induce a stem cell site and differentiate, and people have done that and started to see a better profile. But I guess, I don't know, unless you've got a gene of interest, then it's hard to, I think, have a sort of one-size-fits-all strategy. Even with the mini-gene assays, when we've been able to look at things from blood, they don't perfectly replicate with the mini-gene assay. And that could be through a whole load of different factors. So I think it's a complex world, and the more you look at it, the more complex it's going to become.
Jamie, it's a lovely talk. You're looking for very rare variants in introns in these patients. It would be interesting to know how commonly that occurs in the population generally. So are there some genes in which that happens a lot?
Yep.
A proportion of people have rare things, and others in which it's very rare, because that would also direct your analysis as to which genes you would take seriously. Have you considered doing that?
So this was very stringent sort of criteria. We went with a really stringent frequency in Nomad and a really stringent frequency in the gel data. And a limitation of that is we're only going to pick up things which are potentially completely penetrant with disease. And intronic stuff might not necessarily display complete penetrance, because it's not necessarily an all-size-fits-all, not an all-size-fits-all, that's a stupid statement. If you introduce a new site, it doesn't mean that that's always going to be used. And so I think these particular class of mutations could be more frequent in populations, because you can still have some residual wild-type allele being expressed as well as the mutant. And we've started to see that, particularly with the CFTR variant, that it's not just completely this new transcript which is made. So yeah, I completely agree with what you're saying, that these things could be more frequent in the population than we'd expect, if that was what you say.
Well, I think what I mean is what proportion of people for a particular gene might have a unique, ultra-rare intronic variant.
Yeah.
And if a gene scores highly on that, then you'll find it might not be so important as if compared to if it didn't.
Like a tolerance score, I guess, for intronic.
Yeah.
So yes, the stringency is high, but some genes might have rare things in many people. The MCA4 gene, a large gene, for instance, which we work with. Do you understand?
Yeah, so I mean, size is obviously going to be a big dictator of that, but also the number of motifs. So it's not just the dinucleotides that you need to create a new splice site. You need that polypyrimidine tract. You need the branch point for that actually to become part of the transcript. And so you could, in theory, take the whole of a gene, look at the genomic context, and see how many potential sites there are for these cryptic splice site mutations. I don't know of anyone who has done that, but it'd be an interesting thing to do.
Yeah, I mean, I guess I was just going to take that a bit further. I mean, when people are looking at potentially recessively acting variants, I never understand the idea of having a super rare cutoff. It might not be rare at all, mightn't it? And then if you go and look for a singleton variant or a variant that you've never seen before, I mean, people have got all sorts of rare variants all over the place, haven't they? And I just wonder whether, if you're going down that route is necessarily the best. It never makes sense to me that when you're looking for a recessive thing, you go for something that's super rare, as you would if it was a dominantly acting variant.
So yeah, if you're looking at compound heterozygous variants, in combination they need to be rare. And as Andrew just said, there's genes where, so it depends if you believe the literature or not, but some people say that for USH2A, you could look at anything that's above 0.05%, and you'd never find a pathogenic variant. Whereas in something like ABCA4, you could go as high as 2%, and we're starting to find hypomorphic variants which are as high as 4 or 5% in the population. So yeah, it could be gene dependent, and there are all sorts of additional things that you can start to do, but this is very much a first look at the data. What can we find that's completely penetrant and definitely disease causing?
Yep.
I was wondering if you had any thoughts on how important some intergenic variants might be.
How important what, sorry?
Intergenic variants.
So, yeah, I mean, that's a more difficult thing to do. So how do you define what a gene actually is? Is it between the start site and the end site, or is it all the regulatory apparatus that's intergenic or elsewhere in the genome that is required to regulate it and make sure it's expressed? And without knowledge of what those regions are for each gene, that's a really quite difficult thing to do. So we can go to OnCode, we can get integrated resources that say this bit of chromatin is open, but actually linking that back to a gene is quite difficult. Technologies like Capture-High-C or FORCE-C are starting to show how promoters are interacting with other parts of the genome, and integrating that sort of thing into analysis is something we're doing and something we're really excited about, but actually it has another level of complexity above intronic variants.
Okay, I'm going to use my chair's prerogative to ask a last question. So you were talking about, it seems like you have to look quite hard to find these things, and then you have to do a functional assay. So do you envisage that in the future this will be like a sort of personalised medicine service for rich people that you can buy from a private company? Or do you imagine that you will actually identify more things like consensus sequences that you won't have to confirm with assays, that you'll just be able to bioinformatically predict?
So if you look at where we are now with protein code and stuff, then there's a whole framework as to how you sort of classify a variant from benign to pathogenic. And I can see that existing for non-coding things, and I think that will exist, but it's then you're always going to have a level of uncertainty. So even now with rare or novel missense variants that we know deep down are pathogenic, they get given a criteria of variant of unknown significance. Mm. And I think you'll get to the same standard with intronic variants. In a way, they're sort of, providing you can access the gene, the expression of that gene, they're easier to confirm that it's having an effect, I think, than missense variants might, where you need a specialist functional assay. Actually, we're just looking at the RNA. We just want to know what's being included in messenger RNA. So I in some ways, it's easier than looking at rare missense variants, but I think we'll get to the same place where you'll have things which, on the scale of 1, 2, 3, 4, and 5, where 3 is variant of unknown significance and 4 is a pathogenic variant that you can report, we're going to get 3.5 a lot of the time.
That's encouraging. Okay, well, thank you very much to both the speakers.